Good morning, children. In today's presentation, we are talking about the double fertilization. Double fertilization, it is the process of simultaneous fertilization at two places in angiospermic plant, once in form of the syngamy and another time in form of the triple fusion. Okay. So in case of the syngamy, when we are going to study about the syngamy, there is a fusion of male gamete with the female gamete, and there is a formation of a zygote, and this zygote turn into the embryo. And in case of the triple fusion, when the polar nuclei or the central cell fuses with the male gamete, it forms primary endosperm nucleus. Okay, the ploidy of the primary endosperm nucleus is diploidy, and thus the syngamy and triple fusion simultaneously in the embryo sac is known as the double fertilization. Now we are going to study about the post-fertilization event. After the fertilization occur, then the primary endosperm nucleus, it turn into the endosperm, zygote convert into the embryo, the ovule convert into the seed, and ovary turn into the fruit. Now come to the process of the development of the endosperm. It is the very important question that which one is developed first, endosperm or the embryo. Okay, so endosperm develop first and it is followed by the embryo, and this is because the endosperm has to provide the nourishment to the embryo. So it is very much necessary before the development of the embryo, food must be there. And the endosperm, as we already know, it is the most nourishing substance. Most nourishing substance exists in the nature, and it is the reserve food material and for the nourishment of the developing embryo. As we already know, if we are having any kind of long illness, doctor always suggests to have the coconut water, and this is because the coconut water, it is the best example of endosperm, and it is the best example of endosperm. So after the every long illness, we have to take the coconut water. So we are going to study how this endosperm is formed. So the endosperm develops from the primary endosperm nucleus. The ploidy of the primary endosperm nucleus is diploid, and this PEN undergoes mitotic cell division. Or earlier we know the mitotic cell division has the two phases. The first is karyokinesis and second one is the cytokinesis. Karyokinesis, it is the division of nucleus, and cytokinesis is the division of cytoplasm. Okay, so there is a two types of endosperm. First type is the free nuclear endosperm and another type is the cellular endosperm. In case of free nuclear endosperm, there is only karyokinesis, while in case of cellular endosperm, cytokinesis as well as the karyokinesis, both the process occur completely. The best example of free nuclear endosperm is coconut water, while the cellular endosperm is the kernel of the coconut or the white edible portion of the coconut. In next slide, we are going to study how the embryo is formed. Embryo is developed from the zygote. The zygote undergoes again by the mitotic cell division, and it forms two cells. The first is the suspensor cell and another one is the embryo cell. The suspensor cell, it divides many times mitotically and it forms a long thread-like structure which is known as the suspensor, while the embryo cell, it divides many times by mitotic cell division and it forms a cluster of cell which is known as the proembryo. Okay, the major function of suspensor is it pushes the proembryo inside the endosperm to get the nourishment, and after getting the nourishment, the proembryo divide very fastly and it forms the complete fully formed embryo, and this embryo consists of two cotyledons, plumule and radicle. Okay, and a dicot embryo, you will see here the complete picture of the dicot embryo. In this dicot embryo, you will find the two cotyledons. One is plumule, one is radicle, one is hypocotyle and another one is the epicotyle. So we already know about the function of all these things except the hypocotyle and epicotyle. Okay, so epicotyle, it is the portion of embryonal axis above the level of cotyledon, and it develop into the plumule or the baby shoot, while the later the hypocotyle, it develop into the radicle or the root tip, and the root tip contain a small cap and that cap is known as the calyptra. Now come to the structure of the monocotyledon embryo or the monocot embryo. We know the best example of monocot embryo is the maize, wheat or rice. Monocotyledon embryo consists of one cotyledon only, and it has a small embryonal axis and which is fused with the cotyledon, and this is known as the scutellum. Radicle and root cap, it is situated at the lower end and it is covered by the undifferentiated axis known as the coleorhiza, while the plumule, it is covered by a hollow foliar structure, it is known as the coleoptile. Okay, the best example of monocot embryo is the grass, banana, bamboo, palm, rice, wheat, maize and many more. Okay, now come to the seed. The seed, it is a fertilized ovule and which are the final product of sexual reproduction. It consists of seed coat, cotyledon and embryonal axis.